Welcome back to my daily lectures on New Age Cosmetic White EdTech Healthcare Consultancy. I'm Dr. Bobby. We're now dealing with the Chapter 5 of Molecular Biotechnology, titled the subject in Chapter 5 is Bioinformatics, Genomics, and Proteomics. And the topics under this particular title will be molecular databases, metagenomics, functional genomics, DNA microarray technology, serial analysis of gene expression, proteomics, separation and identification of proteins, protein expression, protein profiling, protein microarrays, protein and protein interaction mapping, summary, and Q&A. Now going to the subject matter proper. Molecular databases, scientific information is generally presented for expert scrutiny, peer-reviewed articles that are published in professional periodicals. However, in the mid to late 1980s, molecular biology journals were into more of DNA and RNA and protein sequences derived from individually cloned genes. So anyone who wished to have conducted comparative analysis of nucleotide or amino acid sequences among related genes or proteins, as well as other kinds of analysis, had the unenviable task of typing out all the sequences from the relevant publications. The GenBank database was established in 1982 in anticipation of the increased availability of DNA sequences. The purpose was the collection, management, storage, and distribution of sequence data. At first, submissions to GenBank were sporadic, but almost complete compliance was achieved when many journals made database submissions a prerequisite for publication. From then on, the database became an integral part of sequence-based research. Initially, access to the GenBank database was through servers that were linked to NSFNet, National Science Foundation Network. By current standards, the original GenBank interface was primitive and download times were interminable from 1990 to 1995. Large-scale projects such as genetic and physical mapping of the human genome, partial sequencing of thousands of complementary DNAs, that is called cDNAs, express sequence tags, ESTFs. ESTs is express sequence tags, and sequencing of entire genomes around requiring additional databases, and the expansion of the existing database for storing and retrieving information. NSFNet was replaced in 1995 by the Internet World Wide Web. The app submission access and especially retrieval data mining of stored molecular information became rapid and easy. The visual aspects of the online improved dramatically, and links among relevant databases were established. Ready access through the internet led to the creation of specialized databases for gene-specific mutations, regulatory sequences, mitochondrial genes, and function-specific to human genetic diseases, protein-protein interactions, protein structures, and gene expression data, and many other types of data. As shown on Table 5.1, types of molecular databases were bibliographic resources, cellular regulation, chromosome aberrations, comparative genomics, gene expression, gene identification structure, gene mutation, gene sequences, genetic and physical maps, genetic disorders, genomic variants, genomic sequences, intermolecular pathways, intermolecular interactions, metabolic pathways, protein motifs, protein sequences, protein structures, protein resources, RNA sequences, and transgenic organisms. Very important. Now, generally, the area of research that generates analysis and manages the mountain of information about genome sequences and all the genes that are described in various cell types and tissues has been designated as genomics. Studies of the entire protein population of various cell type and tissues and the numerous protein-protein interactions have been dubbed proteomics. As new methods were implemented and research targets became more focused, other omics such as metagenomics, functional genomics, that's transcriptomics, metabolomics emerged, where generally the suffix omics implies large-scale whole genome experimentation. And with the analysis of many samples at one time. As a consequence of the high throughput, there is a heavy reliance on computers and computer programs for assembling, analyzing, archiving, and distributing genomic data. Diverse computer resources have been developed to handle the various kinds of genomic information. The amount of information generated from massively parallel experimental systems is huge. For example, since its inception, the sequence information GenBank has been doubling every 80 months. More specifically, after 25 years, GeneRaLBank contained more than 80 billion nucleotide bases from about 80 million sequences derived from more than 100,000 different organisms. There are currently 900 molecular databases and been increasing ever since that range from major DNA and protein sequence repositories such as the GenBank, Ensemble, UCSC, GenBrowser, Genome Database, Universal Protein Resource, and the Ribosomal Database Project that provides genomic and proteomic data to bibliographic and informational resources. For example, OMIM, PubMed, RefSec, and GeneClinics. Clearly, the proliferation of molecular database would not have been possible without either high-speed computers or the programs which develop the means whereby the information can be used by the scientific community. Because of its systemic nature, the extensive use of computers for storage and analysis, molecular data has become known as bioinformatics. So, broadly speaking, bioinformatics is the development and application of computational tools for the submission, storage, organization, archiving, acquisition, analysis, and visualization of biological and medical data. Database contents are routinely accessed through internet sites that have tutorials at this address of browser http://www.worldwideweb.ncbi.nlm National Library of Medicine National Institute of Health governments/education/. Okay, with instructions for accessing the information, explanations of how to use the software tools for sequence similarity searches, gene prediction, and many other kinds of analysis. Most molecular databases are designed to meet the needs of researchers and are not meant for curious visitors. There are, however, many useful websites that provide overviews of genomics, proteomics, bioinformatics, and other related topics. In sum, the adage necessity is the mother of invention, which was coined by Richard Frank in 1624 to 1708, aptly encapsulates how bioinformatics has advanced both genomic and proteomic research. Programs are written to analyze and visualize the experimental data and accessible data. Bases have been developed that are augmented with additional descriptive features, annotations, and linked to other sources of data to combine as much relevant material as possible. Information is now available and accessible that would have been impossible to obtain in the past. Importantly, novel studies can be contemplated that previously were considered impossible. Now about metagenomics. For more than 100 years, the identification of microorganisms and the characterization of the biological functions have required cultivating each strain in the laboratory in the 1990s with the emergence of techniques to extract DNA from environmental samples such as soil and seawater, researchers have began to examine the sequence diversity of microorganisms using the universal 16S ribosomal RNA gene as a taxonomic marker. These studies reveal that less than 1% of all bacterial species could be cultured, and therefore novel genes that might be of considerable interest for basic and applied research were inaccessible using methods that depended on growth of bacteria in the laboratory. Considering the wealth of biotechnologically important genes and proteins that have been obtained from the relatively few culturable microorganisms, the possibility of harvesting useful genes from the much greater number of unculturable microorganisms was exciting, if not daunting. With the development of high-efficiency cloning, robotic workstations that handles thousands of transformed cells, inexpensive DNA sequencing, microbial DNA sequence, database, and bioinformatics resources for processing, standardizing, and storing information, it has become possible to access the genomics of uncultured organisms from environmental and clinical samples. The study of the collective genomes in these samples is known as metagenomics. The primary objective of a metagenomic project is to construct a comprehensive DNA library from all the microorganisms of a particular ecosystem or location as shown in Table 5.2. The metagenomic clones can be characterized in various ways. One tactic entails sequencing the entire library using the shotgun sequence strategy with the aim of assembling contiguous sequences of DNA, contigs, from as many different genomes as possible, and identifying both novel and homologous gene sequence. For example, a massive study that included 50 ocean samples from locations in the North Atlantic through the Panama Canal to the South Pacific yielded 6.3 into 10 raised to 9 bases of sequence. The analysis also revealed sequences encoding potentially novel forms of many proteins, including proteins for repair of ultraviolet, high light-induced DNA damage, and riboscope, ribulose, biphosphate carboxylase, an enzyme that is important for carbon fixation. Sequence-based metagenomic projects are especially effective with microbial communities that have relatively few species. For example, some bacteria are able to thrive on pyrite, iron sulfide, ore sediments, and are associated with extremely acidic runoff from metal and coal mines, acid mine drainage. Not only is this acid water, often pH less than 1, harmful to aquatic and terrestrial ecosystem, but it leaches out environmentally hazardous heavy metal contaminants such as copper, lead, zinc, and cadmium from the ore sediments and mine tailings. The toxic runoff often continues long after the mining operation has been abandoned. Thus, there is considerable interest in learning more about the metabolic pathways of the microorganisms found in these environments and how they survive under such conditions. This information may contribute to more effective control measures for curtailing the production of acid. In one metagenomic study of an abandoned mine site in California, the nearly complete genomes of the two major species, bacterial species Leptospirillum group 2 and Aferoplasma type 2, and partial genomes of 3 other microbes were cloned and assembled. Although more research is required to determine the metabolic dynamics of this microbial community, gene assignments for the different genomics indicated that a rare member of the assemblage, Leptospirillum group 3, plays a critical role. It may be the only organism that fixes atmospheric nitrogen in this environment, and as a result it acts as the primary source of nitrogenous compounds and ammonia for other microorganisms. So, dear friends, as I enter chapter 5 with more details, I request that all of you take notes, make screenshots for the benefit of your studies and writing exams and augmenting your career. Kindly do not make commercial copies for commercial purposes or such matters. Please use it exclusively only for studies, and all credits to the authors of this book, Glick, Bernard, Jack, and Cheryl. Thank you very much for continuing with the subject matter proper. Metagenomic libraries are frequently screened for enzymatic activities to identify novel enzymes with biotechnological potential. As shown in Figure 5.2, selection for growth of transformed E. coli shells on particular substrates, complementation tests, and more are most often simple integrated systems are used. Table 5.2, sources of some metagenomic libraries: abandoned metal and coal mines, Arctic plankton, freshwater lakes and rivers, high-temperature springs and meadows, intestinal microbial communities from insects, humans, and mice, marine sponges, oceans, sediments of all types, sewage, sludge, soils of all types. This flowchart shows the sample, marine water, freshwater, soil, mud, and steins. This is about the construction of metagenomic libraries from various sources. Going back, it shows sample, marine or freshwater, soil, mud, intestine, etc. Then you have concentrate microorganisms by filtration, concentrate microorganisms by centrifugation, extract DNA, clone DNA fragments into a vector, transform E. coli with a metagenomic library. That goes into shotgun sequencing to obtain whole or partial genomic sequences. The other goes into screen libraries for novel genes or operons. Figure 5.2, construction of metagenomic libraries from various sources. Bacteria from marine and freshwater samples are concentrated by using 0.1 to 0.8 micrometer pore size filter, and viruses are concentrated by using a less than 0.1 micrometer filter before the DNA is extracted and then fragmented. Libraries containing small DNA fragments inserted into a plasmid are screened for novel genes, while libraries containing larger DNA fragments inserted into a phosmid, cosmid, or bacterial artificial chromosome vector are screened for operons encoding protein complexes or metabolic pathways. And for these studies, in an example, a metagenomic library was screened for cloned lipase genes growing transformed cells on agar plates that were supplemented with various triglyceride substrates such as tricaprilin and isolating colonies that were surrounded by a clear zone, that is a halo. The halo indicated that the colony produced and secreted an enzyme that digested tricaprilin. Relatively large DNA fragments, typically between 5 and 40 kilobase pairs KB, must be cloned to ensure that all genes encoding proteins in the pathway for catabolism of a substrate often encoded in a polycistronic operon are present in a transformed cell. These function-based metagenomic projects have identified a myriad of enzymes. The availability of robotic systems for maintaining and transferring transformed cells has been extremely helpful for expression-based studies because on average about 10 raised to 4 to 10 raised to 6 metagenomic clones must be screened to detect 1 or 2 positive colonies. However, there is an inherent limitation with the host cell, usually E. coli, for selection schemes that depend on the transcription and translation of the clone gene, computer modeling using codon usage and other transcription translation features from the genes of many different microorganisms suggest that only 40% of the heterologous genes will be expressed in E. coli. Consequently, to increase the likelihood of identifying additional novel genes, broad host range vectors and other host cells are being used for constructing and maintaining metagenomic libraries. Specialized gene expressions systems aid in detecting metagenomic clones that carry genes with certain functions. One example of this type of strategy has been called substrate induced gene expression, that is SGEX screening. As the name suggests, this procedure identifies catabolic genes that are expressed when the promoters are activated in the presence of particular substrates and relies on the cloning of regulatory elements that are often found upstream of the catabolic genes that they control. The system utilizes a vector that contains the green fluorescent protein GFP gene under the control of the LAC promoter P raised to LAC. in a POC-based plasmid designated P18GFP, the cloning site lies between the lac promoter and the GFP gene. DNA from a microbial community is fragmented and cloned into P18GFP. The cells are grown in the presence of ampicillin to prevent the growth of untransformed cells. An IPTG isopropyl beta-D thiogalactopyranoside, which induces the expression of green fluorescent protein from the lac promoter. Cells that produce green fluorescent protein in presence of IPTG are those that carry plasmids without inserts. That is, self-ligated plasmids. Plasmids with inserts do not prevent transcription of GFP from the lac promoter. For example, the insert does not contain a transcriptional terminator, or plasmids with inserts containing promoters that are constitutively active. Transformed cells of interest in this procedure are those that do not produce green fluorescent protein in the presence of IPTG. Table 5.3 shows some of the gene-encoded enzymes identified in metagenomic libraries. They are alpha amylases, antibiotic resistance enzymes, antibiotic synthesis enzymes, example polyketide synthesis, aromatic hydrocarbon catabolic enzymes, cellulases, chitinases, dehydrogenases, 1,4-alpha glucan branching enzymes, lipases, esterases, nitrolases, oxidoreductases, pectin lyases, proteases, vitamin biosynthesis enzymes, and xylanases. This is an illustration to the same effect. Please take a screenshot as you see them. Right? Right, now continuing with the subject. That they carry plasmids with inserts that do not induce expression of GFP under these conditions. To remove cells that produce green fluorescent protein in the presence of IPTG, the transformed cells are subject to fluorescence-activated cell sorting, FACs. Briefly, FACs consists of streaming cells in single file past a laser beam that detects the excitation of a fluorochrome that is either attached to or, as in this example, inside the cell, and sorts fluorescent and non-fluorescent cells into separate collection vessels. Accordingly, with Cytek screening, the cells with green fluorescent protein, which fluoresces green when exposed to blue light, are separated from the cells that do not synthesize green fluorescent protein and therefore do not fluoresce. The green fluorescent protein negative cells are then grown in the presence of a low molecular weight substrate, for example benzoate. The purpose of this step is to identify the metagenomic clones that carry cloned DNA segments that are required for activation of catabolic genes by the interaction of the target substrate, for example benzoate. Regulatory elements, including genes encoding regulatory proteins and the DNA elements that they bind to in the presence of the target substrate, are usually close to the promoter of the catabolic operon, and therefore at least a portion of the catabolic operon is likely to also be present on the cloned segment. If a catabolic operon is activated by the substrate and transcription continues through the GFP gene, then green fluorescent protein will be produced. Consequently, a second round of FACs is carried out, and the cells that express green fluorescent protein in the presence of the substrate are retained. This procedure enables rapid, high-throughput screening of a metagenomic library with various substrates. An initial test of subjects with the groundwater metagenomic library yielded 7 different operons that were induced by benzoate and 2 induced by naphthalene. Bioinformatics systems are being developed to handle the vast amount of data derived from metagenomics projects. For example, an internet resource that has been called KORAH, which stands for Community Cyber Infrastructure for Advanced Marine Microbial Ecology and Analysis, on the following web ad. provides extensive sequence and ecological databases, as well as computational tools for analyzing marine metagenomic details. Figure 5.3 shows the CGEC system for isolating catabolic genes from metagenomic libraries. A CGEC vector designated P18GFP contains the GFP gene encoding gene fluorescent protein GFP under the control of the IPTG inducible promoter PLAC. Top plasmid metagenomic DNA fragments are inserted into the multiple cloning site MCS between PLAC and GFP. If an inserted fragment contains a constituted promoter that is active in the host cell, I'm just magnifying it, and derives expression of GFP or a sequence that does not disrupt the activity of PLAC, example, a sequence carrying a transcription terminator, then the cells carrying these constructs will limit green fluorescence in the presence of the inducer IPTG. If the insert carries a promoter and regulatory elements that are activated only in the presence of a substrate, of interest, example benzoate, and drive TFP expression, then these cells will emit green fluorescence when the substrate is added. ND not determined because cells that express GFP in the presence of IPTG are removed in the first CGEC screening step. B. Metagenomic libraries are prepared for P18 GFP and initially screened using FACS to remove clones that emit green fluorescence in the absence of a catabolic substrate. Non-fluorescent cells are collected, grown in the presence of a substrate of interest, and then screening again by FACS to identify clones that carry substrate-inducible regulatory elements. The inserts from clones that emit green fluorescence only in the presence of substrate are analyzed to identify full or partial sequences encoding catabolic enzymes. Additional experiments may be required to isolate entire catabolic operands. Okay, we now read in a milestone, multiplexed biochemical assays with biological concepts. Courtesy multiplexed biochemical assays with biological chips, courtesy SP Fodor, RP Breva, XC Huang, ACPs and CP Holmes, and CL Adams. Credits to all of them. And quantitative monitoring of gene expression patterns with the complementary DNA microarray. Credits to M. Shenna, D. Shallon, R.W. Davis, and P.O. Brown. Thank you. We're reading those details for you. Fodor et al. Described the basic features of the oligonucleotide microarray fabricated by photolithography. In the words of the authors, recently we have developed new technologies to synthesize and assay biological molecules in a miniaturized format. The method uses light to direct the combinatorial chemical synthesis of biopolymers on a solid support. The identity and location of each biopolymer is known, and its interaction with the molecular binding agent can be measured. These miniature biological arrays or chips can then be used for a variety of multiplex biochemical assays. In the latter article, SPA Fodor et al. Fodor in Science had mentioned that Fodor noted that the applications for oligonucleotide microarrays appear to be only limited by imagination, as said by him. Over a short period, DNA microarrays have become a standard component in the toolkits of both molecular biologists and biotechnologists. Shenna et al. Showed the the utility of a robotic printed spotted cDNA microarray for analyzing gene expression in a large scale. By current standards, a 45 cDNA microarray seems modest. However, in 1995, it was a spectacular demonstration of a new technology. In some, although not necessarily the first articles using or describing multiple array assay formats, the Fodo et al. And Shena et al. Publications showed that DNA microarray platforms had come of age. And the genomic libraries continues. Researchers will also be able to correlate species, abundance, and gene frequencies with environmental and physicochemical physicochemical information, have a better appreciation of the dynamics of marine microbial communities. In a broader context, algorithms have been developed to detect prokaryotic gene sequences to recognize DNA sequences that are specific for in particular microbial species and to distinguish members of known gene families. In sum, the metagenomics approach has begun to reveal an immense amount of information about the vast microbial world that barely a decade ago was considered beyond reach. Functional genomics. In functional genomics, genomics encompasses the study of all features of genomics and individual genes at the DNA level, including mutations, polymorphisms, and phylogenetic relations that are based on sequence differences. Another aspect of genomics that is often called functional genomics or transcriptomics is concerned with patterns of description, either qualitatively to determine which genes are expressed or quantitatively to measure changes in the levels of transcription of genes. Transcription at the whole genome level is assessed as a function of clinical conditions, as a consequence of mutations in response to natural or toxic agents, and different cells or tissues or at different times during biological processes, such as cell division or the development of an organism. One of the aims of gene expression studies is to discover the genes that are up and down regulated under specific conditions, the transcriptome. In the past, the transcription of only one or few genes could be followed at a time. Currently, functional genomics methodology can track the simultaneous transcription of thousands of genes, gene expression fold profiling of either a cell or a tissue sample. The main experimental approaches for determining gene expression profiles are DNA microarrays and series analysis of gene expressions, SAGE. Because of the large amount of data that is generated from these experiments, special computational tools are required for obtaining, storing, and analyzing the results. DNA microarray technology. A DNA microarray, DNA chip or gene chip experiment consists of hybridizing a nucleic acid sample target derived from the messenger mRNAs of a cell or tissue to single-stranded DNA sequences probes that are bound in an ordered arrangement to a solid platform. One type of DNA microarray is constructed by spotting, polymerase chain reaction PCR, amplified cDNA sequences from the mRNAs of a single cell or all or specific sets of the coding sequences of an organism onto a glass slide or nylon membrane. In this case, about 10,000 different probes can be arrayed in a 1 centimeter squared area. This is the description of the figure. Figure 5 for gene expression. Profiling with the DNA microarray mRNA is extracted from two samples, sample 1 and sample 2. And during reverse transcription, the first cDNA strands are labeled with the fluorescent dyes CY3 and CY5 respectively. The cDNA samples are mixed and hybridized to an ordered array of either gene sequences or gene-specific oligonucleotides. After the hybridization reaction, each probe is scanned for both fluorescent dyes, and the separate emissions are recorded. Probe cells that produce only a green or red emission represent genes that are transcribed only in samples 1 and 2, respectively. Yellow emissions denote genes that are active in both samples, and no emission black represent genes that are not transcribed in either sample. Sample 1, sample 2, sample 1 to mRNA, DNA cycle 3, sample 2, mRNA, DNA cycle 5. and continue to matter proper DNA microarray technology. A DNA microarray DNA chip or gene chip experiment consists of hybridizing a nucleic acid sample target derived from the messenger RNAs, mRNAs of a cell or tissue to single-stranded DNA sequences, probes that are bound in an ordered arrangement to a solid platform. One type of DNA microarray is constructed by spotting polymerase chain reaction PCR amplified cDNA sequences from the mRNAs of a single cell or all or specific sets of the coding sequences of an organism onto a glass slide or nylon membrane. In this case, about 10,000 different probes can be arrayed in a 1 centimeter squared area. An alternative microarray system utilizes sets of oligonucleotides as probes, usually representing thousands of genes for one commonly used platform. The probes are synthesized directly in situ on a solid surface quartz wafer by a light-directed process known as photolithography. Thousands of copies of an oligonucleotide with the same specific nucleotide sequence are synthesized in a predefined position, probe cell, or feature on the array surface. For this type of macroarray, the probes are typically 10 to 40 nucleotides, and several probes with different sequences for each gene will be synthesized on the microarray. Longer oligonucleotides, up to 100 nucleotides, can also be used. A complete whole genome oligonucleotide array may contain more than 500,000 probes, representing as many as 30,000 genes. Generally, the design of probe set for microarray depends on the objective of the experiment and the degree of resolution that is required. Computational programs determine probe sequences that are specific for their target sequences are least likely to hybridize with non-target sequences. Cross-hybridize have with the non-target sequences. Cross-hybridize have no secondary structure. Fallback and I repeat, have no secondary structure fallback that would prevent hybridization with the target sequence and have similar melting annealing temperature so that all target sequences can bind to their complementary probe sequences. Under the same conditions, oligonucleotide microarrays may consist of probes that represent an entire genome, a single chromosome, selected genomic regions, or selected coding regions from one or several different organisms. Repetitive sequences are not included in genomic DNA microarrays. This is the depiction DNA microarray, laser scanning to final. Typically for most gene expressions, profiling, experiments that utilize microarrays, mRNAs, is extracted from cells or tissues. Purified, and cDNA synthesized using reverse transcriptase, and they extract the mRNA as a template. Usually mRNA is extracted from two or more sources whose expression profiles are compared. For example, from diseased versus normal tissue, or from cells grown under different microorganisms from different conditions. The cDNA from each source is labeled with different fluorophore by incorporating fluorescently labeled nucleotides during cDNA synthesis. For example, a green-emitting fluorescent dye, CY3, is used for the normal reference sample, and a red-emitting fluorescent dye, CY5, for the test sample. Figure 5.4, after being labeled, the cDNA samples are mixed and hybridized to the same microarray. This is a depiction of the fluorescence image of a DNA microarray hybridized with CY3 dye, CY3A, and dye CY5 labeled cDNA. And now a laser scanner determines the intensities of dye 5 and dye 3 for each probe cell. The probe cells have different colors depending on the amount of CY3 and 5 that are present, and the ratio of red CY5 to green CY3 fluorescence intensity of a probe cell indicates the relative expression levels of the represented gene in the two samples. To avoid variation due to inherent and sequence-specific differences in labeling efficiencies between CY3 and CY5, reference and test samples are often reverse labeled and hybridized to another microarray. In the above, for example, reverse labeling dye swiping would entail labeling the reference sample with CY5 and the test sample with CY3. Alternatively, for some microarray platforms, the target sequence from each source are labeled with the same fluorescent dye, and reference and test samples are hybridized to different microarrays. In an alternative strategy, mRNA is purified with an oligo-DT sequence that binds to the poly-A tail of eukaryotic mRNA and has a short extension T7 primer containing the sequence for the bacteriophage T7 RNA polymerase promoter. The oligo-DT T7 sequence primes the synthesis of cDNA from mRNA using reverse transcriptase. Synthesis of the second DNA strand using DNA polymerase results in double-stranded cDNA. It contains the T7 RNA polymerase promoter. Next, T7 RNA polymerase is used to produce RNA copies, complementary RNA, cRNA, or antisense RNA from the second DNA strand as a template in the presence of biotin-labeled ribonucleotides. This reaction results in linear amplification and biotinylation of cRNA, which is then fragmented into pieces from 50 to 100 nucleotides in length that are optimal for hybridization. After hybridization, the microarray is treated with streptavidin that is bound to the fluorescent protein phycoerythrin. Streptavidin binds specifically to the biotin residues of hybridized cRNA, and hybridization can be detected by emissions from phycoerythrin that are elicited during laser scanning. Because of the vast amount of data generated by microarray experiments, specialized software has been developed to maximize the output of information. Analysis of two-dyed and one-dyed hybridized microarrays are similar. A common method by which information is extracted from two-dye hybridized microarrays is summarized here. Each probe cell of a two-dye hybridized microarray is scanned using a confocal scanning microscope. Following laser excitation of the dye, fluorescence emitted from each probe cell detected at both 532 and 635 nm with CY3 and CY5 respectively is collected through the microscope's objective lens and converted to an electric signal via a photomultiplier tube. The intensities of fluorescence emitted by both dyes for each probe cell, along with background readings for the microarray, are recorded and stored. Background fluorescence is determined by measuring the fluorescence from blank areas where probe cells have not been spotted and is subtracted from the fluorescence intensities measured for each probe cell. Microarrays are designed with internal controls, that is, specific probes that are used to evaluate the reliability of the hybridization procedure and to ensure that the laser scanner performed properly. At this stage, the microarray data, that is the collection of fluorescence intensities of each probe cell, is normalized to correct for variations, systematic errors caused by technical factors that contribute to the fluorescence intensities of a probe cell and enable comparison among the microarrays of an experiment. To minimize errors, multiple probe cells for each gene are included on a single microarray and replicate samples are independently prepared under the same conditions. Figure 5.6, gene expression profiling with an oligonucleotide microarray mRNA is purified with a poly-DT sequence that has a T7 RNA polymerase primer sequence extension. After two-stranded cDNA synthesis, the second cDNA strand acts as a template for synthesis of cRNA by T7 RNA polymerase in the presence of ATP, cytidine triphosphate CTP, guanidine triphosphate, GTP, uridine triphosphate UTP, biotinylated CDP, and biotinylated UTP. The gray circles represent incorporated biotinylated nucleotides. The biotinylated cRNA is purified, fragmented into pieces from 50 to 100 nucleotides in length, and hybridized to an oligonucleotide macroarray. The macroarray is treated with streptavidin phycoerythrin and the probe cells, black squares, are scanned for emission yellow for the biotin-bound streptavidin phycoerythrin. This is a representation of the same total RNA, purification mRNA, first stranded cDNA synthesis, second strand cDNA synthesis of biotin-related cRNA, purification of cRNA, a fragmentation of cRNA, and then the hybridization to streptavidin phycoerythrin staining, scanning oligonucleotide microarray. The replicated samples are prepared under the same conditions and hybridized to different microarrays. Misleading interpretations are caused in part by variations in sample preparation, such as growth conditions, RNA extraction, cDNA synthesis, and labeling efficiencies. Differences in efficiencies for hybridization of the target sequences among replicate microarrays across a single microarray. Variations in the concentrations of probes in different microarrays or unequal amounts of target sequences applied to different microarrays or unequal distribution of targets on a single microarray. Several methods of normalization are used to calibrate the data among replicate microarrays, such as using the fluorescence intensity of a gene that is not differentially expressed under different conditions as a reference point, including spiked control sequences that are sufficiently different from the targeted sequences and therefore bind only to a corresponding control probe cell, and adjusting the total fluorescent intensity for each microarray to a similar value under the assumption that a relatively small number of genes are expected to change under different conditions. One of the major purposes of a microarray experiment is to identify genes whose expression changes in response to a particular biological condition. The response to a biological condition is determined by comparing the fluorescence intensity for each gene, each probe cell, averaged among replicates under two different conditions, and calculating the ratio commonly expressed as an n-fold change. For effective comparisons, the raw data of the dye emissions of each probe cell of a microarray are often converted to log2 ratios. The sign indicates the dye with the higher intensity. Generally, positive log ratios represent more CY5 and CY3, and therefore greater expression of the gene in the test sample than in the reference sample. Negative values of more CY3 and CY5 indicate a lower level of expression in the test sample than in the reference sample. The log ratios for all probe cells are compiled into a table called an expression matrix. Microarray analysis is also used to identify genes that are co-expressed under different conditions or over a period of time, with the goal of determining which gene products function in a given way. A number of computational strategies are available that organize the data into related groups, clusters. For a clear presentation of the clustered data, ranges of log ratio values are assigned arbitrary colors. Usually black is designated for a log ratio of 0, dark to bright red for increasing positive log ratios, and dark to bright green for decreasing negative log ratios. In other words, red is frequently used to denote gene overexpression, and green to denote underexpression. A visualized representation of a clustered microarray is called a gene expression profile, where the rows represent the reordered genes and columns represent either conditions or samples. The gene expressions profile in figure 5.7, determined by microarray analysis, clearly shows that different genes are transcribed in patients with cirrhosis of the liver than in healthy individuals, and in patients with ethanol-induced cirrhosis than in those with cirrhosis induced by hepatitis. The table shows the feature genes, CY3 intensity, CY5 intensity, CY5 by CY3, log2 CY5 by CY3. This is a depiction of the illustration of gene expression profile of a cirrhotic liver tissue. Columns 1 to 7 and 8 to 15 are liver samples from patients with ethanol and hepatitis C virus-induced cirrhosis of the liver, respectively. Each patient's sample was compared to normal liver tissue The cluster consists of 2,965 genes. The asterisk denotes patients with severe cirrhosis of the liver. And the next figure illustrates gene expression profile of lymphocyte-specific genes from cirrhotic liver tissue. Columns 1 to 7 and 8 to 15 are liver samples from the patient shown in figure 5.7, methanol and hepatitis C virus induced cirrhosis of the liver, respectively. Each patient's sample was compared to normal liver tissue. The cluster consists of about 70 genes. The asterisk denotes patients with severe cirrhosis of the liver. And the C virus, hepatitis C virus. Moreover, there is a difference between the genes that are turned on during advanced ethanol-induced liver damage and those in patients with less severe ethanol-induced cirrhosis. No such distinction is evident among individuals with different severities of virus-induced cirrhosis. In addition, information about the transcription of genes that contribute to a particular pathway or cellular activity can be extracted from a gene expression profile. For example, genes that are transcribed during lymphocyte proliferation and activation are highly expressed in virus-induced liver cirrhosis and to a much less extent in ethanol-associated cirrhotic samples. The importance and pervasiveness of DNA microarrays cannot be overstated. In 2007's example, there were more than 13,000 published journals articles used this technology or describe methods for enhancing data analysis. Clinical applications for DNA microarrays are being developed. U.S. Food and Drug Administration granted permission in 2007 for the first commercial diagnostic assay based on DNA microarray. In this case, a 70 gene expression profile, MomaPrint, courtesy them, distinguishes between patients with breast cancer that is likely to migrate to other sites, metastasize, and those whose cancer has a low risk of metastasis. And the reliability and reproducibility of microarrays with different formats and from different laboratories have been major concerns. However, standards for both running microarray experiments, analyzing the data, have been proposed by a number of international groups which should alleviate these problems. Finally, it should be noted that in addition to gene expression studies, DNA microarrays are used to determine the binding sites of DNA binding proteins. Example, chip-on-chip assays which use chromatin, immunoprecipitation, CHIP, or chip, to identify proteins bound to a DNA microarray. The sites where the transcription of genes starts and stops, and many other aspects of genome architecture. This research has shown that a much larger proportion of the eukaryotic genome is transcribed than was previously thought. A number of genes have multiple start and termination sites, some of which are hundreds of kilobases from known sites for many genes. Both strands of many genomic regions are transcribed. Splicing occurs between RNA molecules, and some transcription factors bind to dozens to sites scattered throughout the genome. In short, whole genome microarray analysis has revealed greater complexity in the processes that control transcription in a eukaryotic organism that could be predicted through smaller scale transcriptional analysis. Serial analysis of gene expression. Unlike DNA microarrays that rely on hybridization and signal detection, SAGE uses recombinant DNA techniques to clone randomly linked short sequence of cDNA prepared from extracted cellular mRNA that can be efficiently sequenced to identify expressed genes. Polyadenylated mRNA is captured by an oligoid DT sequence that is labeled with biotin and attached to a streptavidin-coated magnetic bead. Double-stranded cDNA synthesized from the purified mRNA using a reverse transcriptase to synthesize the first strand of cDNA with the oligo DT primer and mRNA template, and then DNA polymerase to synthesize the second complete complementary strand. A strong magnet is used to retain the magnetic beads with attached cDNAs during successive treatments and washings. The cDNAs are cut with restriction endonucleases NLA3, which recognizes the sequence and cuts outside the GC base pairs. leaving a 3' GTAC extension. This lecture will be over in a minute or so, and the rest of it will continue in the next lecture. I'll just wind up in a couple of words now. Since NLA3 cuts on average 1 in 256 base pairs BP, there is high probability that each cDNA will have at least one NLA3 recognition site. Right, so we are winding up on this now, and just to mention, the cDNAs are bound to beads. Each NLA3 cut site that is closest to the 3' end of a cDNA is retained, and all unbound fragments are washed away. Nomenclature developed for SAGE NLA3 is called anchoring enzyme. So that's it for now. Take notes, take screenshots, make your study clear and good, and get back to me on any doubts that you have on the comment section. I'll get back to you accordingly on the concerned video. Thank you, best wishes, good luck.